Good morning, good afternoon, good evening, and welcome. I am Amy Wagner, Global Sales Director with Technibus Crystallization Systems. Today, we are happy to bring you our latest webinar on crystallization strategies in the quest for increased yield and chiral purity in collaboration with APC and presented by Dr. Andrew Byrne with APC. As a reminder, we are recording this webinar and I expect it will be sent to you via email early next week. We do have the audience muted, but we will entertain questions at the end. Please send these via the chat function or directly to info@crystallizationsystems.com. The webinar will last 45 minutes. Again, it is great to see many of our current customers joining us today who are users of our crystallization technology and quite familiar with Technibus crystallization systems. For those of you joining us for the first time, Technibus Crystallization Systems is a Dutch company based in the Netherlands. We are focused on developing innovative instruments for applications including solubility screening, crystallization process development, and formulation applications. We have more than 500 systems in all areas of the world in markets that include pharma, agrochemical, and fine chemicals. Featured in today's webinar are both our Crystal 16 and Crystalline. Both instruments are multiple reactor systems that combine heating, cooling, stirring, and transmission measurement within each reactor. The Crystal 16 is specifically designed for screening applications, including generating phase diagrams for solubility curve metastable zone width determination, solvent screening, polymorph salt, and co-crystal screens. The Crystal 16 with 16 reactors operates at a 1 ml scale, so you can quite easily generate solubility curves on up to 4 different solvents overnight with minimal starting material. Once prepared with your solid state roadmap created from the Crystal 16, the Crystalline will help you optimize your crystallization or formulation process with extra analytical capabilities, such as adding in situ cameras so you can now see your crystallization or precipitation as it occurs. Our integrated cameras or microscopes provide particle size information that allows you to develop the correct process for form and habit control, and watch precipitation as it occurs in your formulations. Additionally, the Crystalline can utilize Raman probes that you may already have in-house to follow polymorph transformations and chemical reactions. The Crystalline with 8 reactors at a 5 ml scale provides the information you need to optimize and to troubleshoot your crystallizations. So if you finish this webinar and think, boy, I would really like to have one of these instruments in my lab, we certainly would be very happy to work with you to answer any questions on the systems or their capabilities for your applications. You can contact us at info@crystallizations.com or very easily through our website. I know we would be happy to help you. And now I'm very happy to introduce our speaker today, Dr. Andrew Byrne. Andrew joins us from APC in Dublin, Ireland, where he's a senior research scientist. He graduated from University of Limerick and has his PhD in pharmaceutical organic chemistry from Trinity College, Dublin in 2014. Andrew has been working in the world of PAT and process development since he started at APC in early 2015. His projects have included work in particle engineering and crystallization, route scouting, reaction optimization, and flow chemistry. And Andrew, thank you for joining us and sharing your knowledge. You should now be able to share your screen.
Good afternoon, everyone. My name is Andrew, and today I'm going to present to you on APC's crystallization strategies and their targeted application in the quest for increased process yield and chiral purity. I'd first like to thank Technibus Crystallization Systems for this opportunity to share with you here today, and I look forward to our subsequent discussions at the conclusion of the presentation. Presented is a general overview of what I'm going to cover today, a brief background of stereoisomers, chirality and their applications in active pharmaceutical ingredients, an introduction to ternary phase diagrams and why they are so relevant to enantioselective crystallization. I then plan to present two case studies. Case study 1 will be ternary phase diagrams as a tool for process definition. Case study 2 will be enhanced process understanding and how it leads to yield improvement. In each case, I will present a problem statement, outline our methodology and development steps, and give key outcomes, conclusions, and recommendations. And I will then conclude with some learnings and acknowledgements. Presented on the current slide is an overview of Apc and Our Partners. Apc is a CRO, or Contract Research Organisation, based in Dublin, Ireland. Apc employs over 140 scientists and engineers working across a range of modalities from small molecule to large molecule. We partner with large biopharmaceutical and pharmaceutical companies around the world to accelerate the delivery of their medicines to the market through process development and ultimately accelerating the delivery of that medicine to the patient. Through a synergy of chemistry, biology, and engineering, we offer value by enabling rapid process feasibility, process definition, development, and characterization, so our partners can accelerate the development process. Ahead of case study number 1, where we'll look at the development of enantioselective crystallizations, I would just like to do a quick refresher on the concept of chirality within active pharmaceutical ingredients. With altered chirality, compounds may possess altered physiological and pharmaceutical effects. For example, the desired enantiomers for the presented APIs exhibit favorable therapeutic effects, such as analgesia, antitubercular, antiarthritic, and anti-nausea. However, their chiral analogues possess undesirable effects, such as toxicity, blindness, and teratogenic. Chiral small molecules in 2019 generated large revenues, with Lipitor, Plavix, Seretide, and Crestor sales in the billions of dollars. Of course, the ideal scenario for the synthesis and purification of enantiomers is 100% selectivity. However, the reality can be far from this, with many classical synthetic routes generating racemic mixtures, or mixtures of your desired and undesired enantiomer or enantiomers. There are channels for optimization within resolution strategies, prochiral substrates, and catalyst loadings, leading to increased enantiomeric excesses. Chiral chromatography is also possible, but has a high associated cost. The crystallization unit operation, when correctly designed, can be harnessed to reduce the development time and increase process yield and chiral purity. To continue our high-level refresher, I have selected a few key terms and properties for this glossary. Chirality is a geometric property of a molecule. A chiral compound is non-superimposable with its mirror image. Enantiomers are chiral compounds that are non-superimposable mirror images. Diastereomers, in contrast, are non-superimposable and not mirror images. A key observation here is that enantiomeric pairs have identical physical properties, while diastereomers have different physical properties. This property will be clearly evident during the solubility design space construction and solid phase characterization, and this will be further illustrated over the following slides. The characterization of the solid phase is a key step during the design of enantioselective crystallization. This allows us to understand the crystal lattice arrangement and whether our two chiral compounds crystallize as a racemic conglomerate or solid solution. A conglomerate crystal lattice arrangement is most favorable. This characterization of the solid phase can be achieved with binary phase diagrams or ternary phase diagrams, both presented on the slide. During binary phase diagram characterization, a DSC is used to monitor the melting point of solid mixtures of chiral compounds. During ternary phase diagram construction, we monitor solubility of solid phase mixtures and plot this on our ternary phase diagram, where the eutectic point is the highest solubility point of the mixture. These diagrams are typically symmetrical for enantiomers, but one-sided for diastereomers, as a result of the aforementioned different physical properties. I will now introduce our first case study number 1. Ternary phase diagrams is a tool for process definition, where we will apply this aforementioned theory. In this high-level process flow diagram, we see that a hydrogenation reaction is worked up following a number of charcoal treatments, filtrations, distillations, and multiple restories. Very much a lab-scale process. There were a number of processing issues. This was extremely long. It took over a week to go from hydrogenation to isolated API. There were a number of inconsistent key outputs, such as enantiomeric purity, yield, assay, variable heavy metal content, and a very, very inconsistent purging of rhodium, iron, and zinc. APC were tasked with measuring the API solubility of this conglomerate system, determining the effect of heavy metals, if any, on the solubility, to develop an optimized enantioselective crystallization to isolate the desired enantiomer, and then to demonstrate this on 10 gram and 100 gram scale. Process establishment is a key phase in any process development project, allowing us to assess and understand the key quality attributes of the current state here at the APC laboratories. By applying APC's data-rich experimentation ethos, we gained key information around each of the individual unit operations for assay, purity, and heavy metal content. It was noted that with each unit operation, chiral purity and assay weight percent increased, but this was not the case for the heavy metal purge. In fact, it was observed that both charcoal treatments provided little in the way of heavy metal purge relative to the IPA water reslurries. This was an early indicator to the team that these unit operations could be altered or indeed removed. As part of the process assessment, an extensive mass balance was carried out around each of the individual unit operations to characterize all the process rich and waste streams. A selection of the data is presented on the current slide. Here we see a loss of about 10 to 11 mg per gram of our desired material to the post-reslurry liquor when studying our liquors with the reverse phase method. However, the true story can be observed through the normal phase chromatography, where we see that a 50-50 mixture of our desired to undesired is lost to this liquor. A key learning here is that the selection or development of the correct HPLC method for solubility measurement is vital. Selecting a method that separates both enantiomers allows individual quantification and can avoid a common development pitfall. APC has developed an in-house platform for early phase solvent selection and crystallization design. This approach harnesses in silico computational screening tools to predict solubility, relative solvate propensity, or how likely something is to form a solvate with the solvent that it is in, and impurity purges. This allows us to screen a large panel of potential solvents, allowing the selection of a short list of candidates for experimental validation via 2-point solubility and proof of concept crystallizations. This helps us to minimize time, cost, and effort during the development process. Presented on the current slide are the outcomes of one such computational screen, utilizing Cosmo ORS and Dynachem. Here, 27 potential crystallization solvents were ranked based on relative solubility of our desired enantiomeric compound. Good solvents or poor antisolvents were identified during this screen, such as DMF and DMSO. And also, good antisolvents or poor solvents, such as octane or heptane, were also highlighted. As a solvate was not the desired outcome of our crystallization, the screen allowed us to eliminate a number of potential solvate-forming solvents, such as formic acid and THF, from our experimental screen. There are two commonly used in-house methods to construct Ternary phase diagrams. Today, I will share both of them with you. The classical isothermal method can be used paired with HPLC sampling. In the case of our process, this involved the synthesis of the undesired enantiomer through modification of the catalytic and reaction conditions. Excess solids are charged into a known volume of your crystallization solvent, allowed to equilibrate, and the supernatant is sampled post-hold. The desired and undesired concentrations are collected, utilizing our normal phase HPLC in terms of mg per gram, and converted to weight percent. This allows the data to be plotted directly onto our ternary phase diagrams. The data presented in the bottom right-hand corner is an example of one such set of data from our case study. You can see that a simple eutectic composition is obtained at the ratio of 50-50, as this is an enantiomeric system. An alternative method for ternary phase diagram construction leverages crystal 16 clear point determination. Here there's a twofold objective: to characterize both the solid phase and to generate our solubility design space or roadmap. Here we produce a range of known solid mixtures or ratios of the desired to undesired, adding known quantities of this mix to a known volume or mass of solvent and collecting solubility. Crystal 16 clear point is obtained therefore for the mixture at each composition and solvent matrix. Personally, I would recommend spot checking here to determine if there's any degradation or racemization, which can occur in the case of enantiomers during the collection process, that may affect your solubility data. The clear point data are solubility points for the mixtures at given temperatures are then graphed as per normal. Isothermal cross sections relative to the process conditions are then taken. Solubility is interpolated and converted to weight percent to allow then for the construction of our ternary phase diagram. Lines connecting solubility at the same temperature on a ternary phase diagram are known as isotherms. And in the example given, we have a simple eutectic conglomerate system at 50-50, which will be again typical of our enantiomers. A quick experiment for the verification of a simple eutectic is presented on the present slide. In this particular experiment, saturated solutions of our desired and undesired enantiomers are individually prepared. Their isothermal solubility is presented here to the left and right of the ternary phase diagram in figure A, as measured by HPLC. To each liquor, excess solid of the alternative enantiomer is then added, and post-equilibration the liquor is sampled by HPLC. In a simple eutectic system, in isothermal conditions, we should see convergence on the same eutectic solubility, regardless of the starting enantiomer provided. That is, of course, given each enantiomer is in excess. And this was the case for our system, as displayed here in figure B. At this point of our early phase process development, we were armed with a key understanding of the solubility design space for our system and how to construct it. We now wanted to address one of the key objectives for our project to determine the impact or potential impact of residual heavy metals, zinc, rhodium, and iron, and the effect they may have on API solubility throughout the process. In order to do this, we isolated solid at various stages of the process and collected solubility using our normal phase LC method. As you can see, at the midpoint of the process, with elevated levels of metals, we have similar solubility at 3 temperatures. So we decided to stress this and compare the solubility of our crude solid post-hydrogenation to the purified material. And here you can see large levels of rhodium, zinc, and iron. Again, we observe similar solubility, and when plotted onto our ternary phase diagram, the data sits nicely on our isotherms. This data contrasted with the initial hypothesis of the project, but also highlighted how important targeted and informed solubility collection really is. Our potential process roadmap is outlined by the construction of our ternary phase diagrams. A proof of concept crystallization was executed utilizing a 70-30 mixture by weight of desired to undesired enantiomer in 50-50 IPA water. As with all process development here at APC, we employed the relevant PAT or process analytical technology to maximize the yield of data from each of the process conditions. Here we utilized in situ FTIR and FBRM to track our seeded cooling crystallization. Following dissolution and effective seeding, we can observe a steady desaturation, indicated here by 0.4, until about 30 degrees where a secondary nucleation event occurs, and this is picked up by our in situ FBRM and FTIR. This experiment was also followed by HPLC, liquor, and solid sampling. In this crystallization system, the initial desaturation corresponded to the desired enantiomer with the concentration of the undesired remaining stable in solution. Until, of course, event number 5 at 30 degrees, where we see the secondary nucleation and subsequent desaturation of both enantiomers in a eutectic ratio from solution. Transferring this data to a ternary phase diagram, we can visualize and quantify our process from seeding to desaturation, secondary nucleation, as we cross the phase boundary, where we build up supersaturation of that undesired enantiomer, right down to the final isolation point at our eutectic composition. To conclude, in this case study, ternary phase diagrams were employed as a key tool in early phase process definition for chiral crystallizations. They enabled the development of a scalable, shorter, more robust process whereby a crystallization and recrystallization were used to replace a number of facile and impractical unit operations. The developed process improved yield, improved enantiomeric purity and assay purity, and also offered excellent heavy metal purging capability. For case study number 2, I hope to demonstrate how enhanced process understanding through data-rich experimentation can reap dividends for our partners. In this commercially manufactured oncology asset, our partner produces a mesylate through a reaction with MSA in acetone water. Following clarification, this is purified and isolated by crystallization. On plant scale, the average yield of this process was 81%. A full mass balance for this API was not available, and hence the fate of the remaining potential yield was unknown. Process development opportunities found within the project had to lie within strict global filing limits. APC was tasked with enhancing process understanding with the ultimate goal of reducing waste and increasing yield. The current state process was characterized intensively on transfer to the APC laboratories. Presented on the current slide is the PAT-enabled crystallization assessment. The current state crystallization process was a seeded cooling crystallization from MSA, acetone and water. Particle morphology and growth was tracked by in situ PVM and FBRM across dissolution, seeding, desaturation, and during the whole period prior to isolation. The product was routinely isolated with rod-like morphology with adequate filtration properties. The proven acceptable ranges defined by the global filing of this product are displayed at the top left of this slide. The set points for the standard process were at the median values within the proven acceptable range for MSA, water, and acetone. A mass balance executed early in the process development cycle indicated that up to 14% of the total crystallization yield was lost to the post-crystallization liquor and washes. Opportunities for data intensification often present during process assessment. As this process was a cooling crystallization, the post-crystallization solvent matrix was equivalent to the initial composition at dissolution. Hence, at the end of each crystallization presented today, the true process liquor solubility was also collected via Crystal 16. To truly understand a crystallization, you must construct the relevant solubility roadmap. By doing this, we can quantify supersaturation, metastable zone width, and determine the operations which contribute to the largest yield precipitation. The global filing indicated strict ranges of proven operating conditions for the process strain. This included volume limits on MSA, acetone, and water, as previously mentioned. From previous experience here at APC, we know that small variations in solvent matrix composition can lead to significantly altered solubility, and hence yield. As a proof of concept, a number of artificial solvent compositions were targeted to investigate the ranges of operation. The solubility of the system was found to vary slightly within the artificial solvent systems, all within the limits of the PAR. As a result, it was decided to further investigate this with some true process streams. I have previously mentioned the set point conditions for the global process filing and related ranges for acetone, water, and MSA. In this proof of concept study, the true process liquor composition was stressed to assess the impact, if any, on process yield, process mass balance, and liquor solubility. Please note the solubility presented here was obtained from true process liquors post-crystallization, maximizing the yield of process information within a reduced number of experiments. It was noted that in all conditions with decreased water and MSA, solubility of our API was increased, but conversely, solubility was decreased with high volumes of water and MSA relative to the standard liquor solubility. The solubility collected, in fact, was only half the story. The corresponding mass balances for the three varied true process liquor compositions are presented on this slide. As expected, the process isolated yield was significantly impacted through the targeted manipulation of the acceptable solvent matrix. And through this, we modulated solubility prior to isolation. In this manner, it was possible to identify a route to yield improvement by reducing liquor losses. The detailed definition of the future state process is an important stage of any process development. In an ideal world, we would like to maximize the yield at all costs. However, that would mean working at the very edge of the proven acceptable range. And as we know, this is not desirable. Our new recommended set points must be within the PAR and allow for some variation. Within the current example, our final optimized condition afforded an isolated yield of 90.8% with equivalent PSD form and purity, within ranges that were slightly inside of the PAR. This process was characterized intensively through a synergy of offline and online analytics. You may ask, so how did this optimized crystallization process perform on scale-up after we transferred it back to its manufacturing site? Here we have a run chart with 27 batches with the yield itemized batch by batch of the API pre and post crystallization optimization. We can see that the mean yield for the process is increased by approximately 7% post APC development activities. To conclude my presentation for today, we covered a brief background in theory on stereoisomers and ternary phase diagrams. 2 case studies were presented. In case study 1, I discussed ternary phase diagrams as a key tool in early phase enantiomeric crystallization development. It was shown that with these ternary phase diagrams as roadmaps, with integrated PAT and targeted experimentation, shorter, scalable, and more robust processes can be developed. In case study 2, enhanced process understanding enabled by data-rich experimentation was harnessed to access yield improvements in a commercial product. The development strategy utilized was discussed that allowed a rapid yield improvement of 7%. I would like to finish with an acknowledgement for the fantastic group of scientists and engineers at APC, without whom this work would not be possible. I would also like to thank you so much for listening, and I would be happy to take questions now or to follow up later by email. Thank you.
Thank you, Andrew, for your presentation and case studies. One question, I did see that you were quite a bit using our Crystal 16. I did want to ask you, how much time is it taking you typically to perform the solubility measurements?
So in terms of our typical process development workflow, if we execute a crystallization that's not anti-solvent addition, post isolation, you will have that liquor remaining, and it's actually a really good habit to almost collect the solubility of that mother liquor. We would typically, isolate on the day and then run solubility in that mother liquor on the evening of. You would then arrive into the lab, and after an overnight hold, with your Crystal 16, it's very well automated, so we have that data the next day. So we find it quite useful.
Excellent, thank you. We have some great questions and feedback from our viewers. One question that I have for you coming from our viewers is, how can we determine monotropic and enantiotropic relationships of 2 or 3 anhydrous forms?
I suppose it's about breaking it down into the individual. So we've had a number of more complex case studies of diastereomers and enantiomers here at APC, and it's about really just understanding the solubility roadmaps that correspond to each one, breaking it down into the individual systems first, and then making it more complex as you develop the roadmap.
All right, I have another question. I want to keep us on schedule, but I do have one other question. When doing the solvent screening, how do we determine that coming out compound is a solvent or not? A solvate or not, I'm sorry.
In theory, so we have, as part of our in-house kind of platform technologies for this, we have an in silico screen. So we have Cosmo RS, which allows us to determine the solvate propensity. And that is a theoretical indicative screen, which allows us to kind of, with high precision, identify whether it is likely to become a solvate or not after crystallization. Obviously, if you use your crystalline and in combination with our solid suite of analytics here, so your DSC or TGA, you can identify the solid state of pretty much any solid form that's produced out of these kind of mock crystallization, small scale crystallizations that's executed on a crystalline. And obviously you can track that by Raman as well.
All right, thank you. I have one more question for you since we have time. Using a ramp of 0.1 degrees C per minute takes a really long time to get duplicates and most likely needs to run the Crystal 16 overnight. Any HSE issues ever faced?
We risk assess each and individual operation here at APC, and I believe it's covered under the risk assessments that we have here for our general operation of the crystalline and Crystal 16, but we've had no health and safety problems with running the Crystal 16 or crystalline overnight.
Excellent. Well, thank you again, Andrew, for sharing your knowledge with